All right, it's my pleasure today to introduce Associate Professor of Psychiatry in the Indiana University School of Medicine, Dr. Susan Sangha. Welcome, Dr. Sangha.
Thank you, Steve. I'm very excited to be here.
Great, yeah, and so we're going to talk a little bit today about a recent paper that Dr. Sangha has published in psychopharmacology, and then also some of the background of the work and some broader implications of it. The paper is Interaction of Stress and Alcohol on Discriminating Fear from Safety and Reward in Male and Female Rats, and this is Hackleman et al., 2023 in psychopharmacology. So Dr. Sangha, could you tell us a little bit about what the main questions you asked in the study?
Right. So the idea for this study was kind of a long time coming for my lab, as I've been thinking about how safety and reward learning overlap for several years now. And we've spent several years studying safety cues and how they can downregulate fear when they're presented alongside fear cues. So if you present a fear cue and a safety cue at the same time, like a compound cue, a conflict cue, however you want to think about it, we often see a reduction in fear behavior. Almost always in male rats. And in a lot of our work, females do not suppress their fear to the same extent as the males do in the presence of a safety cue. However, some of our recent unpublished work that we're doing right now is not always there. The sex difference, it does seem to at least partly be explained by number of trials and the time between trials even. Still needs to be worked out, but I wanted to mention that it's not always there.
So are you saying that the females are basically, is it fair to say they're more cautious or sort of more wary of you, that they like, even though you're telling them it's safe and this cue has recently told us that it's safe, they just sort of don't believe you or something? Or is that how
Yeah,
you
so
think
when
of
you
it?
look at the original data, just sort of at surface value, you're like, well, are the females not understanding the safety cue? Do they not understand that the safety cue is supposed to be a safety cue? Or do they understand it? They're just not showing the fear reduction. We think it's the latter because we've done a lot of different control experiments. A lot of that's been reported in a paper a couple of months ago in Neurobiology of Learning and Memory, where we compared more carefully the males and females in this task. And there's a lot of evidence to show that the females are learning the inhibitory value of the safety cue. It's just that there may be over expectation of threat, perhaps, in the females that is resulting in equal fear when the safety cue is on board, whereas the males show the reduction. So I do think they understand what the safety cue is. It's just the inhibitory value of the safety cue is not enough to counteract that fear cue that's being presented.
Interesting. Okay. And then, yeah, so tell us a little bit more about this specific study and
Yes. So that was all under stress-free and alcohol-free conditions that I just described. But of course, we're interested in the effects of alcohol and stress on this type of safety learning. We know clinically that PTSD can often be comorbid with alcohol use disorder, and that if individuals with this comorbidity are often more resistant to treatment. So I've always been interested in getting into alcohol with our safety paradigm and combining it with stress. So a few years ago, we finally were in a position to able to do that. And our first paper looking at alcohol and stress on safety cue discrimination is this psychopharm paper, which was led by an undergrad in the lab, Abby Hackleman, who did her honors thesis. This was her honors thesis, this psychopharm paper, basically.
impressive.
Yeah. Sometimes, things just work out, right? And then, sometimes they don't for some thesis projects. So this is a purely behavioral paper designed to assess the effects of prior stress and alcohol history on a simpler safety cue discrimination task. So for the discrimination task used in this specific study, it was a little simpler than what I described earlier in terms of we don't have the compound fear plus safety cue in this paper. We simply have the reward cue paired with sucrose, the fear cue paired with foot shock, and then what we're calling a safety cue, but it's just a CS minus paired with nothing. 3 cues that they discriminate amongst. And both males and females under stress-free conditions are able to discriminate beautifully in terms of their fear behavior and their reward-seeking behavior. So that you have a nice high level of fear to the fear cue, that's appropriate, high level of reward seeking to the reward cue, which is also appropriate. And then neither behavior during the safety cue. So both males and females are doing that under stress-free conditions. When you introduce a stressor, I'm getting ahead of myself. Let me back up. There's a lot going on in this paper. So that is the basic task we used. And what happened for all animals at the very beginning of this study is that they had 5 weeks of intermittent access to alcohol. And that meant in their home cage, they had an alcohol bottle, 15% alcohol, and a water bottle 3 times a week for 24 hours at a time. That's a pretty standard intermittent access model used in the field. So they did that for 5 weeks in their home cage with no other manipulation. And both males and females kind of escalated their drinking over the 5 weeks. That's also normal. Overall, the females were showing more alcohol consumption than the males. That's also fairly typical in rodent studies, that the females drink more, just at baseline.
And this is not like, is there like any sugar or anything like that in the water to get them interested in, or are they just purely?
No, we thought about that, but then we were using sucrose as a reinforcer later on, and we decided to try it. Without it, it worked reasonably enough for us to do a study. And I should say also, these are long Evan rats. I don't think it said that, who typically don't drink a lot. And so later on, when we look at the effects of stress, I think that actually did us a favor by having a kind of a lower baseline, because then there's more room to move in escalation, right? Anyways, that's maybe not true. I think that was it helped us a little bit, maybe. Okay, so after 5 weeks of that, all animals had that. And then half the animals underwent a single episode stressor. And the stressor we used was a single 90-minute session where they were exposed to 15 unsignaled, unpredictable foot shocks at an intensity of 1.0 milliamps and 1 second duration. It's quite strong. It's meant to mimic sort of a traumatic event. And later, when they go through behavioral conditioning, we change the foot shock intensity to half of that. So it's 0.5 milliamps for half a second, which is what we use in all our studies. So very strong stressor. And then the control animals were put in the same boxes for the same amount of time, but no shocks. And this was done in a particular context. Let's call it context A, with stress. Everything else later, the behavioral conditioning, is in context B.
Gotcha.
All right. So, so that means overall for this publication, all animals had the same access to alcohol, the same behavioral conditioning. The only difference was whether or not they were exposed to the stressor. Described, they escalate their drinking. And so we were hoping to see after stress that there would just be this immediate escalation in drinking, which did not happen. We replicated many times. It doesn't happen, at least not that first week. So after the stress, we continued the intermittent access in the home cage for 1 week, and we saw absolutely no changes in drinking behavior. So 1 week of that, and then they moved into behavioral conditioning. So our behavioral conditioning paradigm for this paper was basically 2 weeks long. The first week was just simple reward conditioning, auditory cue with sucrose for 1 week. All the stressed animals for those first 1 or 2 sessions showed very much reduced sucrose seeking and some elevated freezing to the reward cue, which was an auditory cue. But by the end of the week, it went away. So by the end of the week, all groups looked the same in terms of their reward seeking behavior to the reward cue. Then after that, there was basically 1 week of this cue discrimination task where we have the same reward cue with the same sucrose outcome. And now we're introducing the fear cue shock and the CS minus trials in the same sessions. And once we start to reintroduce that shock, the stressed animals and reduce their reward seeking for the remainder of the experiment every day. Very low reward seeking and huge amount of generalized freezing to the reward cue, much more than the prior week when they just had reward conditioning. It was probably the most generalized freezing I've ever seen in any of my studies. And both the males and females were showing this in the stress groups. Let's see what else was there. Okay, so there was a lot of generalized freezing, and throughout behavioral conditioning, they're still going back to their home cages and have access to the alcohol and the water. Except now that it's continuous access because we're doing daily behavioral sessions. And it was too complicated to try to keep an intermittent access in that situation. So we moved them to continuous access to both alcohol and water. And when we correlated the generalized freezing during the reward cue to how much alcohol they drank in the home cage, we saw a really nice positive correlation, meaning the more the animal froze during the reward cue, the more they drank in the 24 hours right after that session.
Interesting.
Yeah.
So is it like are they drunk during the task? Or is it just
No, we
afterwards
don't have that kind of high levels of drinking. And also all the data we show is averaged for a 24-hour period. When we look at them, people have described drunk rats to me. We do not have drunk rats.
Okay, so they're just kind of abuse, or not, maybe not abusing, I don't know, sipping on the alcohol, getting a decent amount, but they're not necessarily like wasted in the task. But you're interpreting this more as maybe like a consequence of the task. In other words, like they are sort of compensating for the task by drinking more, that kind of thing. Is that right?
Yeah, I think so. That is consistent with both the males and females. I will say with the males, the drinking prior to the session also correlated with the freezing. So it's hard to tell which way it went with the males. The females were clear. It was only how much they drank after the session was what was correlating with the generalized freezing. So that might be something to see if that replicates and disentangle that.
So would you expect that if they were, let's say you just gave them a bunch of alcohol before this kind of task, like what would happen? Like, does it change their behavior? I mean, I'm asking, I don't think you did this experiment, at least certainly not here.
No, we didn't do that. I'm kind of glad they weren't drunk because then you have movement issues you have to deal with, and we measure freezing. So I think we may have false positives on freezing if they were drunk. You know what I mean?
See. Yeah. So if they're like totally wasted, then they're just laying on the side of the cage and it
well,
thinks
yeah,
it's freezing, but in fact they're just passed out or whatever.
Well, at least that's easy to see in a video. Like,
sure, yeah. But like a lower dose of alcohol, I wonder, one could imagine that alcohol makes an individual more bold or something like that.
Maybe, or it may also interfere with the stimulus discrimination. They may start to like, which one was it again that does the shock?
Interesting, yeah. Oh wow, that's a whole another set of another set of questions. But okay, so then maybe did you have any other findings that you wanted to mention?
I think I went through all of them. So overall, like I think in the paper we pitched it that there are more pronounced effects in the females. Like when you go through the figures, it looks more like eye-popping in the females. But overall, we saw basically the same effects within the males and within the females, and that stress, increased generalized freezing, and increased alcohol consumption for both sexes.
Right. Cool. Okay. Yeah, and I was curious about that. You mentioned something that I found kind of interesting. It's kind of stuck in my mind was that you happen to use a particular model. It sounds like you haven't done a lot of alcohol studies in the past. Is that fair to say?
That's very fair to say.
Okay. And then you just happen to use a model in which you didn't sweeten the alcohol. I know that the people in the alcohol field have these discussions all day long about what's the best way to get animals to drink and so on. And I don't know that much about it, but it sounds like the model you happen to use in these particular rats, et cetera, caused something that was actually fortuitous. You just made a choice, and it was kind of low levels of drinking, maybe in the grand scheme of things, but also it was convenient because you might see increases, which you did. So I'm curious if you could comment any about these kind of like surreptitious or these sort of like this like I don't know what the right word is here, but these happy accidents that sometimes occur in science. Like it sounds like it occurred here. I've certainly seen these in my own lab as well. And then also, of course, there's unhappy accidents that can also occur sometimes. I wonder if you can comment on anything along those lines.
Yeah, unfortunately, there seems to be more unhappy accidents in general, right?
Yeah, I guess most
unhappy accidents, or maybe I'm just, maybe I'm doing over expectation of threat here. Yeah, so one thing that happened with this paper, I said we were doing the simple cue discrimination, but most of the time our lab is interested in doing the conflict cue, like how can the safety cue downregulate fear when the fear cue is there? And that was supposed to happen for this paper. So we did the 1 week of reward conditioning. We did the 1 week of simple cue discrimination. And at the very end, there was supposed to be just a one-off test session to test for condition inhibition, where we would take that CS minus, presented at the same time as the fear cue to see if it downregulated cued freezing, and also presented at the same time as the reward cue to see if we can downregulate sucrose seeking. That was the initial intention. And, things happen. And I guess we were lucky that it was the very last session. So we still had a usable data set regardless, but all of our control animals were given the wrong metassociate program that day. And so we don't have any of the data from the control animals, but we do have data from the stressed animals. And I can tell you for these animals in this study, so this was not in the paper, this condition inhibition test, but when you take that safety cue, the CS minus, and present at the same time as the reward cue in the stressed animals, we actually see a really nice downregulation of that generalized freezing. I told you we had this huge increase in generalized freezing during the reward cue. It's like on average, it was like 60%. And it went down, I forget what it went down to, but it was a significant downregulation in both males and females. And as far as I know, I don't think anyone has really shown a safety cue can downregulate generalized responding. And so I'm really excited about this and moving forward with this, that a safety cue can downregulate both cued freezing as well as generalized freezing, because we know generalized freezing and fear is a big problem with stress disorders. There's a lot of CS minuses in the world, or should be treated as CS minuses, neutral cues that we have overgeneralized responding to. So if a safety cue can downregulate that, I think that'd be really powerful, and to figure out what those neural circuit mechanisms are. So that's what I'm kind of working on now as a new grant idea.
I see. Interesting. Yeah. So so if I'm understanding correctly, like, in PTSD, somebody has a traumatic situation, and obviously it's actually, I mean, it sucks to have a traumatic situation, but you should learn about the traumatic situation. Don't get yourself in that situation again, try to avoid it, etc. But the problem becomes a lot of times people have extra fear that sort of comes from things that are not predictive of a traumatic situation, that are actually innocuous or day-to-day living kinds of things, and they end up with terrified responses because you're overgeneralizing. Is that basically the idea?
Yes, yes. And it seems like maybe our safety cue will be powerful enough to downregulate that.
So how would you use that? Is there a way to, have you thought about, like, I don't know, are we talking about psychotherapy? Are we training people on specific cues that sort of maybe can counteract the fear that they feel under certain circumstances? Is that kind of the implication?
Yeah, I haven't thought too much about the therapeutic, like behavioral therapy on this. Because, well, everyone kind of knows that exposure therapy is sort of the standard right now, which is basically fear extinction. And the idea would be, well, would you introduce safety cues during a fear extinction type treatment? And there's, a little bit of data in the field in rodents that show that this might actually help. But I don't know yet.
Interesting.
Because the way it's complicated, because the way we always train things in our lab is the CS minus is always there during the fear cue as well. We don't train it separately. And I feel like for it to be really learned as a safety cue, there has to be this element of danger for it to really be learned as a safety cue. If you're in a safe environment and you're given a CS minus, I don't know if that will turn into a safety cue. I think there has to be some element of danger in the environment. I haven't formally tested that. I would like to, but I know there's some other people who think along the same lines.
And maybe this is a naive question, but what if you were to train, like, explicitly a reward type of cue under those circumstances? Let's say you have the person in the clinic and they're doing their extinction therapy, which is, I think, largely cognitive in nature, or maybe it's at least overriding a Pavlovian association is the idea. You're experiencing it over and over again without the trauma part, and it kind of goes away with extinction formation of the extinction memory. But is this a crazy idea to think, like, maybe what if you just trained them, like, here's another cue for some candy or whatever, or something like that? Is that the same thing, not the same thing as what you're talking about?
If fear extinction is happening, I think it'd be probably the same problem as what I said. If you have a CS minus in there, separate from the fear cue, and even if you put the CS minus, that neutral cue, on top of the fear cue during fear extinction, that original fear memory may still be preserved because it's different enough from the fear plus safety situation. It's complicated. It all depends on like how the original fear memory was formed, I think.
I see.
I think the same thing would happen with, reward. We have done is do mixed fear and reward extinction at the same time. We don't continue giving the sucrose, but what we have done in a lot of our studies is after the safety paradigm, then extinguish the fear cue as well as the reward cue, and they both extinguish super fast compared to if you had just fear conditioning, let's say. So I think there is something beneficial about going through the safety cue discrimination, that there are other possibilities in the world maybe about how a cue could, be associated with other things. I've never actually published that, but I've always noticed that the extinction seems much faster
Hmm.
when I have safety training versus if you just did fear conditioning to a single cue when no other cue is available, and then you try to extinguish that. Yeah.
I see. Interesting. So you're thinking about this more along the lines of not so much like implementing safety cues in the therapy context, but more understanding what is the difference between someone who doesn't overgeneralize and does overgeneralize, like who can learn or not learn these safety cues. Like a person who doesn't have a PTSD presumably can learn the safety cues and go about their day, but still stay away from bears or whatever, you know.
Yeah, we're kind of doing the latter. So I do have a collaboration with a clinical neuroscientist up at
Hmm,
Marquette, Jacqueline Fitzgerald,
hmm,
who's basically kind of adapted our rodent test to humans. And we do have a publication with just, like undergrads, healthy undergrads with no clinical disorder, but she is recruiting PTSD individuals right now. So the idea is that stimulus discrimination is going to be different in the PTSD versus a healthy control, and that hopefully we will see some neural correlates in the fMRI. And we're looking at the PFC amygdala functional connectivity. So this might be a good time to talk about what we've seen in our rodents in terms
exactly.
of circuits, because that's kind of why we chose the PFC amygdala functional connectivity for the human study. We know in our task, a lot of our neural circuit manipulations have been done in males because, like I said, we typically see that the females don't show the safety effect. And most of the time we're trying to interfere with the safety effect to see what the neural circuits are involved. So if they don't already show the right behavior, then what are we manipulating? So until now, we've really just focused on the males. So we do know that the infralimbic input into the central amygdala is necessary in male rats to show safety expression. And this fits pretty well with the fear extinction literature as well. We know we also manipulated infralimbic to BLA, which was not necessary for already learned safety, but we do think it's probably really important during the acquisition phase as they're learning the cue discrimination. But once it's all learned, the actual expression of the already learned safety, like it's moving from the BLA to the central amygdala, is what our current hypothesis is. And that is also largely consistent with what's happening with the fear extinction literature too. So yeah, so we're very interested in PFC amygdala connections.
Right. Yeah. And is it like all, so you mentioned central and basal amygdala. Was it basal or basolateral?
Depends
We're
on which
distinct,
study.
right?
The DREAD study
Depends on
was
the study. Okay, okay.
basolateral
But
amygdala. Yeah.
gotcha. Yeah, because I do kind of wonder about those distinctions sometimes, like central and basal anything is are obviously different in terms of the basic sort of GABAergic or glutamatergic cortical like
Mhm.
structure of them. But yeah, I do wonder about those lines sometimes. Like I think a lot of these divisions may not be maybe more sort of historical in nature than functional in nature actually. Um, but yeah, so you told us a little bit about the prefrontal inputs to the amygdala. Is there any other circuitry that we're aware of in terms of either safety or fear circuits that are relevant to these topics?
From our lab in this task, those are the ones we've worked out. We are starting to also look at prelimbic and nucleus accumbens and the whole circuit and how they're all interconnected. Don't have any real findings to report on that, but Jacqueline Fitzgerald and I was actually just doing the proofs this morning. We have a new review in press where we
Cool.
talk about neural circuits across animals and humans in this condition inhibition type task. And there's a lot. There's the retrosplenial cortex is important in both humans and rodents for safety. And obviously the PFC and amygdala I've already mentioned. Yeah, mostly there's a lot of different cortical areas that cross over from humans to rodents and hippocampus as well. So
Hmm.
yeah.
It's so I'm sort of I do a lot of stuff with aversive motivation these days, but I'm really a kind of a reward person, addiction researcher, whatever. Um, and so I wonder if you could comment on what, if any, relationship there is between what you're defining as safety signals and like reward. So is this like a type of reward? Is this like a negative reinforcement related thing, or what is there a connection there? Or are these just separate things that are sort of playing out in obviously complex behaviors, but they're ultimately separate, or is there some commonality there?
Yeah, this is a good question that we're trying to tease apart. So we do a lot of recordings in our labs in awake behaving animals during our safety task. So we have some publications with single unit recordings in the basal amygdala and the infralimbic region. And both of those regions have neurons that discriminate amongst these cues. We also, so that there's, a neuron that cares just about the safety cue, for example. But we also have large populations of neurons, both those regions, that respond the same to the safety and the reward cue, and not like to the fear cue at all. So it does prove actually that there's some kind of overlap in neuronal signaling with the safety and reward cue. And yeah, that was really exciting for me because when I started doing the safety work, we all knew like the safety cue is going to suppress fear, but it was still kind of not a novel idea that safety and reward learning overlapped, but there wasn't a lot of neurobiological evidence yet that they did. And so seeing these neurons that were coding the safety and reward cues similarly in multiple brain regions was like very exciting. That was when I jumped up and down in the lab to see when I saw those neurons. So now we have like these subpopulations of safety specific neurons and safety reward overlapping neurons. And our current grant under stress-free, alcohol-free conditions is trying to figure out, are they projecting to different subregions? So for example, we're testing the idea that maybe this infralimbic to central amygdala projection is more about the safety specific signaling, whereas the nucleus accumbens may come on board when it's an overlap with the reward signal. So we're doing a lot of projection specific recordings right now in order to tease that apart.
Interesting. Yeah, these are, I'm really excited by this, by the what you just described, those kinds of studies and the studies you've been doing for some time, looking at conflict and more complex situations than, the sort of standard fare of like either just train a rat to do something great or something terrible. And really the world's more complex than that. And it turns out the brain cares about that a lot. And there's a lot of mysteries probably that still need to be unraveled. So I'm really excited to be watching this work out of your group and others. And I was also just especially intrigued by, another paper that I saw of yours, that there's just tremendous overlap in the populations here in terms of which are responding to, purely fear stimuli, purely reward stimuli, purely, and then the mixture of those. There's a lot of complexity here.
Yeah, and actually, as we're getting more complex, I mentioned earlier that we don't always see sex differences. This is actually, so when we're doing our recordings, some of our newer recordings, we bought this Denscopic system to do single cell calcium recordings because it's easier to do longitudinal recordings in that situation because we want to look, across time, an individual neuron, how it's changing. Now with the alcohol and stress project, we can easily follow them through alcohol, stress, and behavioral conditioning. But anyways, with the single cell calcium imaging, it uses an LED to do the recording, and you can't leave that on for 2 hours, which is actually what our behavioral session is at baseline, is a 2-hour session with the number of trials and the ITI. So we had to make adjustments in the number of trials and the amount of time between trials to pare it down to something like a 40-minute session, maybe give extra sessions. And now we're getting a much bigger range in how good safety is in both the males and females, in a way that the males and females are actually not different as a group average. So we have good learners and poor learners within both the males and females. And it's about right now, under our current conditions, 50-50 split.
Interesting.
Now, even though the behavior basically looks the same under these conditions, I am a firm believer that the neural mechanisms leading to it are probably going to be different in the males versus females. So the behavioral endpoint looks similar, but the neural pathway to get there, I don't think is. Maybe some mechanisms are. Like there's probably some global universal mechanisms regardless of sex, but I think there's going to be some specific differences. What those are, I don't know yet.
We will find out. But yeah, these things pop up all the time. And I think your work is really fascinating in this regard, because if you think about things that there should be sex differences in, you think of, I don't know, mating behaviors certainly is the obvious one, maternal behaviors in rats. And when you think about things that there shouldn't be sex differences, and in fact, often aren't sex differences in, you think of like, don't die, or like, don't freak out when you shouldn't, or go get the thing when you're supposed to. Like, these are definitely common problems for males and females of mostly any species. And yet, as you say, the evidence just keeps piling up that somehow male and female brains, on average at least, tend to compute these problems and emit behavior that is equivalent on the back end via different mechanisms. So like, why is that? It just always keeps occurring to me, and I keep thinking about like, why would evolution have such a situation where we have two, like a perfectly good way to solve something, and the other sex solves it in another way? Like, why would it be that way? What does that tell us about the brain, if anything?
Yeah, this is a really good question. And thinking about ethological differences, it's sometimes I like thinking about it, sometimes I don't think about it. Because uh what was I going to say about the oh, one thing I was going to say, something that always gets asked of our lab is about estrus when we think about the sex differences. So recently we showed that we looked at estrus. There's no correlation with the female behavior in our safety task with estrus. And we were pretty well powered for that. So I think that we can just put that aside for now, that it's not an estrus, simple estrus effect at least. So that leaves connectivity and maybe balance of activity is one idea that I've thought about, especially in the PFC. In the fear literature, it's kind of well known that the prelimbic and infralimbic kind of have these opposing roles on expression and regulation of fear, where prelimbic, so overall, we want to say it really simply and simplistically, that prelimbic kind of drives fear expression, whereas infralimbic is more involved with suppression of fear. But it's going to depend, if one input and they're both going through the amygdala to doing this. So if you can imagine they're both sending projections to the amygdala, but one's stronger versus the other, the balance of activity between prelimbic and infralimbic may be a little altered for some reason in the males versus females, so that we do get the overexpectation of threat in the females. Yeah, and everyone talks about, well, maybe the females need to be overexpecting threats so that they go back to the nest. I don't know if that's really true, but that is one thing that comes up often in these discussions.
Interesting. Yeah, that's there's a lot of mystery for sure. And do you think that like one thing I always like to also think about, and it's a hard question, I'm not expecting an answer, but what does this tell us about humans? You know what I mean? When it comes to these kind of questions, is there like a lot to learn about in terms of like the either sex, on average sex roles in responding to threats, or in terms of brain mechanisms or anything like that?
Yeah, so I think one way I approach it is, so we have the female rats that we're seeing over expectation of threat at baseline, even under stress-free conditions. And then you would expect that to be increased under stress conditions. Then we move over to humans, and I told you we published this one paper with healthy undergrads on our task in humans, and there were no gender differences, which actually I'm quite happy about, that women and men are able to learn it equally. But when we move into PTSD, maybe that will be different. The level of lack of safety, maybe it will be different, although I don't think other laboratories have seen that when they do peer safety conflict tasks, but perhaps our setup will be a little different because we have a reward element in there as well. It's higher stimulus discrimination load for our task. But basically, if you see threat over expectation in both human women and female rats, perhaps those neural circuit mechanisms will overlap with that similar behavioral endpoint.
Mhm.
When we start thinking about escape behavior and, aggression and sexual behavior, it's really, I think, quite maybe different in rodents versus humans, and it might be harder to find those conserved mechanisms, perhaps.
Yeah, there's definitely a lot of outstanding questions in this territory, that's for sure. But it's just fascinating that it's so universally found that there's just a lot more sort of differences in male and female brains and in individuals of within the sexes as well than we sort of commonly appreciated in neuroscience. And I think there's a lot more to discover here for sure, that if we want to do anything about this knowledge and actually implement some treatments for some of these disorders and that kind of thing.
We have so much to do with, continue to include females, obviously, in all studies, in a thoughtful and rigorous way.
For sure, for sure. Great. Okay. Well, this has been a really interesting conversation. I think I really appreciate, you taking the time here and also, moving the field forward in this creative way with this line of studies. I think it's really, taking some of the learning theory stuff and turning into really interesting research that is relevant to psychiatric. Not that it wasn't interesting just for its own sake, of course, but also with relevance to psychiatric disorders and things like that. It really is, I think, moved the field forward. So I appreciate all your efforts, including in the journal Psychopharmacology. So with that, I'll thank you again for your time, Dr. Sangha. It's been a pleasure.
Thank you so much. This was fun.